Hello, everyone. This is a mandatory collaborative group project by students in Dr. Steve Alice's Genetic 2400 class at California State Polytechnic University, Pomona. We have edited together a video recording of our respective parts due to scheduling issues and appreciate your understanding. We hope you enjoy hearing about our project as much as we enjoyed making it for you.
Hello everyone, my name is Davis Mabry, and along with the other members of our Genetics 2400 group here at Cal Poly Pomona, I am going to explain our project involving genetic modification, utilizing recombinant DNA. Now we know that a lot of people get spooked when the idea of genetically modifying organisms comes up, but hopefully by the end of this presentation we can show that this process has the potential to be beneficial for society, and in this case the environment as well. When selecting our project topic, a main theme came up of climate change. While this issue is not new, it has become increasingly prevalent both in the academic community and in mainstream media. It has become clear that if changes are not made to deal with the rising atmospheric carbon dioxide levels, we will see catastrophic events globally. If you need an example of this, look no further than the record-breaking wildfires that are currently plaguing our state of California. The question then became, how do we combat climate change using techniques in genetics? What we came up with was the idea that if we could genetically modify trees to grow faster, we could quickly replant woodland areas that have been decimated by human activity and natural disaster, helping to maintain or increase photosynthetic activity in relation to carbon emissions should, in theory, mitigate some of the effects of climate change. Using this as a hypothesis, we decided that our goal would be to insert the CLE41 gene found within a plant called thailcrest into the poplar tree. The CLE41 gene has been shown to increase speed of growth in thailcrest. Therefore, we believe that it is possible to increase the rate at which the poplar tree reaches maturation by implementing this gene into its DNA. This is likely possible because the entire genome of the poplar tree is known. If our experiment is successful, we believe that further work can be done on other species native to all terrestrial biomes in order to help offset climate emissions on a global scale.
This is Allison Nguyen, and I will be explaining why we chose the organisms we did, the gene, the plasmid, as well as explaining the first few steps of the method. So the organism that we will be genetically engineering are the poplar trees, Populus trichocarpa. We chose this tree because it's a very widely planted, fast-growing tree whose whole genome sequence is available. A gene from another plant, the Arabidopsis thaliana plant, will be inserted into the poplar trees. We chose the Arabidopsis because it has a very fast life cycle, produces numerous self-progeny, has very limited space requirements, and has a very small tractable genome that can be manipulated by genetic engineering much easier and faster than any other plant genome. So the gene will be the CLE41 gene in the Arabidopsis. This gene was chosen because it controls the vascular cell division of the plant while retaining the xylem orientation of the cell division. When this gene interacts with PXY, a receptor kinase in itself or in any other plant, it leads to increased vascular growth while still maintaining the cell orientation. So our overall plan is to insert the CLE41 gene into the poplar trees. So then when the PXY-CLE pathway in the poplar trees is manipulated, the gene will increase the number of cells in the vascular bundles of the tree. More vascular bundles would mean more conduction of water and nutrients in the primary growth stages of the tree, while with the CLE, it will retain the orientations of the cells in the tree. So for our methods, the CLE will be inserted into a plasmid PDGB1 alpha 2 plasmid. We chose this plasmid because it is expressed specifically in plants and it does have an empty vector. Yet the plasmid does lack a promoter, so we had to find an appropriate promoter for the CLE41 gene before it can be inserted. It was found that the IRX3, which stands for Iroquois class homeodomain 3 promoter, would be suitable because this promoter is important in early development of plants and is tissue specific for expression in the xylem. So after the initial insertion of the promoter and the development of the plasmid, the CLE41 of the Arabidopsis would be isolated and used as a template in PCR for amplification using primers that target the CLE41 gene. After amplification, the gene will be inserted into the PDGB1 alpha 2 plasmid. Once inserted, the introns do not need to be removed since this is a plant-to-plant transfer.
Hello, Professor Alice. This is Crystal Barba, and I was in charge of the methods and materials as well. Picking up where Allison left off, after the CLE41 gene is amplified, it is then inserted into a plasmid, and that plasmid is introduced into our vector, which is Bacillus cereus. Now, we chose this vector because it is a growth-promoting rhizobacterium, which means that it's very common in the soil, and it is a bacterium that helps the roots grow and absorb nutrients. After the CLE41 gene is inserted into the Bacillus cereus, we wanted to give it an antibiotic resistance, so we chose kanamycin. Now, once all the surviving bacterials are left, we will then isolate that plasmid, and the Bacillus cereus would be able to transfer the new DNA or the new gene into the poplar tree. This would cause the vascular bundles to increase within the plant because the PXY and the CLE pathway has been changed and amplified.
Hi, my name is Juliana Quinones, and I will be discussing the ethical considerations surrounding the genetic modification of the poplar tree. There are 3 specific issues I would like to point out. First and foremost, there is the issue of genetic modification in general. Some believe that we should not have our hand in altering the genetic makeup of organisms, while others have no issue with it. Unfortunately, this is a highly debated topic that seems to have no clear answer nor solution. Moving on to the possibilities that could happen as a result of genetically modifying the organism. A main issue is the possible detrimental effect that this experiment could have on the organisms surrounding it. To see what effects the genetic modification and rapid growth of this tree would be, I did research on possible ways to limit the risks. I found that a confined field trial could be a possible solution. A confined field trial includes planting the trees in a greenhouse at first with no other organisms present and watching the effects. Then, moving other organisms into the greenhouse to see if they would be disturbed. If we see no harmful effects to the other organisms that were moved in, we would slowly introduce the modified poplar tree to a natural environment. I would also like to consider a legal obstacle we must jump through to follow through with this experiment. To genetically modify an organism, we must get a permit from the United States Department of Animal and Plant Health Inspection Services. To attain a permit, we must submit information to this department regarding specifics of the experiment, such as what genes we are inserting or researching, the purpose of our experiment, along with other details to ensure the conditions this experiment requires is safe for the environment and everybody involved.
Hello, my name is Lalo Saavedra, and I will be discussing the future direction and practical implementations of our research. After going through extensive testing and obtaining appropriate clearance, we would test in small controlled areas first in poplar trees, and then in large-scale open fields. In the initial testing in small controlled areas, we hope to develop a consistent high success rate of enhanced growth with the introduction of the plasmid with the CLE41 gene. If our experiment was successful, the next step would be to insert this plasmid into thousands of poplar trees in an existing natural ecosystem. If we can rapidly grow trees that absorb more carbon dioxide at higher rates, we can effectively reduce the amount of carbon dioxide in our atmosphere. In addition, younger trees can absorb carbon dioxide more quickly while they are growing, rather than older trees. So if our experiment is successful, we can then analyze if the results are beneficial to offsetting greenhouse gases, and then from there, possible implementation into other photosynthetic organisms to enhance growth in a wide variety of plants. In addition, it will be necessary to extend this research to other organisms in order to replant biomes with their respective native species.